really prefer to have a chalkboard, not a chalkboard, whiteboard, where I can write on, draw phylogeny, and so forth, and that just makes things so much easier. All right, so we'll just get right into the lecture here. But before I do that, let me back off a little bit on the worksheet from yesterday, okay? For on the ferns, one of the things I was brought to my attention at the end of the lab yesterday was that I'm not referring to the photo atlas for the ferns. For the bryophytes, yes, there's a lot of references to figures in the photo atlas, but not in the for the ferns. And there's a reason for that, because the photo atlas just doesn't have very good, it has pictures of sporophytes, but it really doesn't show much of anything else. Therefore, I didn't reference it. What we normally had in the lab, we have these posters that are on about ferns and fern life cycles, and even moss life cycles that we put out. And that was really the reference the students went to. So as you do your analysis and examination of the ferns, what you need to use is figures 2 and 3 that I put into the pre-lab notes, and also use lecture notes, okay? Because I'm pointing out in lectures a lot of the things I'm asking you to label on the figures that you have for lab exercise 5, okay? Just that's how it's going to go. All right, and so I'm going to go over some of that stuff now. So let me go back, start this, getting my act together here. And share the screen. And we move on. Okay, even though I got a little bit beyond this, I'm not going to review the chlorophyta because we covered that pretty well. But one of the things I want to say is that one chlorophytes happen to be phylogenetically more closely related to land plants than they are to other chlorophytes. And so right now the chlorophytes are the chlorophyta. It's paraphyletic. We're still going to accept it like that and just go on because it requires some more study to be able to divide it up. And furthermore, that would create several other clades that we'd have to talk about, and I want to minimize the number of clades you have to learn. Just remember, though, that whenever you see on any exam or quiz we're talking about and designating chlorophyta, there will be no apomorphies for it because it is not monophyletic. And furthermore, that you need to recognize that it is a paraphyletic group, all right? Other than that, we're moving on. So in the land plants, transition from freshwater to land, we're pretty certain that the transition was not from the marine habitat to land, but from freshwater to land. Most of the chlorophytes happen to be freshwater, okay? And furthermore, a lot of fossil areas where we find the earliest multicellular land plants seem to be around lakes and rivers and not marine habitats. Once again, to emphasize as we go through all the plants, one of the things that we'll see through all their life cycles as we keep emphasizing life cycles is keep this idea of alternating generations in mind, because this is exactly what the major structures that we'll be looking at that differentiate these various groups of plants are there, okay? Gametophyte and sporophyte. Gametophyte alternating with sporophytes in their life cycle. All right, so this is the phylogeny, and I'll be referring back to it. This is the complete phylogeny. So I don't know if in your notes, if I have eliminated some of these apomorphies that I show on this tree. If so, then don't worry about it because we'll be coming back to this phylogeny. You'll be able to fill it in. So this is the land plants. However, what I've put in here is one group of chlorophytes that sometimes is taken out of the chlorophyta and put by itself. This is the charophyta. Charophytes. These are multicellular or colonial, and they appear to be some. You'll still find them treated as green algae. Okay, but in the future, there will probably be in a group all of their own, but it's a very well supported monophyletic clade. I'm not going to put any apomorphies here for it, but what ties this group of green algae to the land plants, okay, the land plants is all this right here, okay, with the bryophytes, the ferns, gymnosperms, angiosperms, that's the land plants, is that two apomorphies that tie the charophytes with the land plants. And that is that they have, and I've already mentioned this, they produce spores. That's one of their reproductive structures, besides gametes. They have gametes as well. But the spores contain a very hard polymer called sporopollenin. And this sporopollenin is only found in the cell walls of spores of both charophytes and the land plants. And so that's something that's shared there. Something else that's also shared is cytoplasmic connections between cells. And I've mentioned these before, probably in Biology 101A, plasmodesmata. But just to review once again what a plasmodesmata happens to be, it's sort of analogous to gap junctions in animals. So if we have here a plant cell with its cell wall, and I'm not going to draw the cell, well, this is the cell wall, is that normally inside of the cell wall on each side, of course, you have the plasma membrane of the cell. And therefore, for transport material from one cell to the next requires going through two plasma membranes. But what we have here is a very narrow channel, cytoplasmic channel, okay, a aqueous tunnel going from one cell to the next. So these are called plasmodesmata. Now this allows small things like ions and so forth, maybe simple sugars, but definitely nothing larger than that. Proteins, organelles cannot pass through this, but it allows these to be able to move from one cell to the next. This is also what makes the spread of viruses in plants a real big deal, because viruses that happen to invade one cell have an easy passageway from one cell to the next through these plasmodesmata. So these are two apomorphies. You don't find them anywhere else, right? Okay, and that links the charophytes with the land plants. Now, the monophyly of the land plants, which we're calling here plantae, is well established. There's several apomorphies here. For one, heteromorphic alternation of generations. Now, this I want to emphasize here heteromorphic alternation of generations, because there are some green algae, such as, and it's also close to plants, called ulva. And it's further down here from the charophytes, but it's a multicellular green alga. And it's not the only one, but they have alternation of generations too, where they have sporophytes and gametophytes, but morphologically, they look the same. You can't tell them apart, okay? Unless you extract their chromosomes, look into their cells, you can see that one generation happens to be haploid, the other generation happens to be diploid, but otherwise they're morphologically identical. Here, the gametophytes and sporophytes are morphologically distinguishable. They look very much different from each other. And so so here the apomorphy is not simply alternations of generations. It is heteromorphic alternation of generations. And that's very important to put on your exams. That little distinction is a very big difference. Furthermore, they have a waxy layer on the surfaces, cuticle. It's a wax that's secreted to prevent water loss. The multicellular gametangia, okay, gametangium. Yesterday in lab introduced the two different types of gametangia. The gametangium is a that's in terms of analogous words, gametangia in plants is sort of analogous to gonads in animals. Now here we're not differentiating between what type of gonads, but more specifically what we have as gametangia in plants is archegonia and antheridia, okay, which is analogous to ovaries and testes in animals. Now, these are not homologous, absolutely not. It's analogous, okay? So they have a multicellular gametangium with sterile tissue, okay, cells that protect it okay, from drying out and so forth. And so when I use this word sterile, it's not used in the same sense that we use in microbiology, free of any living organism. It means that it is not reproductive, okay? It isn't not, those cells do not engage in meiosis or mitosis to form, since this is gametangent, they don't engage in mitosis to form gametes, all right? They're there for protection. Once we have fertilization, and it takes place in an archegonium, always in an archegonium, egg, sperm come together to form the zygote, and the zygote will divide by mitosis, becomes an embryo, but that embryo is a sporophyte. The embryo is a sporophyte, but it is retained in the gametophyte for some time, and it's dependent on nutrition from the gametophyte, and this is an apomorphy as well. Okay, so these are the apomorphies for plantae, all right? So and we'll come back to this phylogeny as necessary. Now, there's 3 different lineages that we collectively call bryophytes. Now, if we treat this as a clade, which it is not, then it would be paraphyletic, because one of these groups, and I believe it's moss, is more closely related by phylogeny to the rest of the plants. But that's adding, traditionally, we've always taught in botany classes or botany coverage of plants in general biology, they usually separate the non-vascular plants from the vascular plants. And I'm going to follow that here for simplicity, okay? Because we already have too many clades to get to know. And when we get to the animals, there's a lot more than what we have here in plants. So we're collectively calling these bryophytes. And I really should have this bryophyte with a little b, okay? So there's 3 different groups here, very similar in that they do not have vascular tissue, okay? But that's not an apomorphy, that's a plesiomorphy, because we don't find vascular tissue in the green algae. And the best outgroup to polarize characters in the plants, of course, would be the green algae, especially Charophyta, okay? That'd be a good outgroup. So hornworts, liverworts, moss. We're mostly going to concentrate on moss because this here is the biggest group of these 3. And so you notice here, I put this together as a polytomy. I'm not going to differentiate how their phylogeny actually is, although it is and has been worked out. We do understand it. So just collectively, these in a Linnaean classification, each one of these groups are given a scientific name, and I'm not going to worry about the scientific names for these groups, but they would be treated as at the phylum level in a Linnaean classification. They're just moss, hornworts, liverworts, okay? And of course, they have no vascular tissue. What is vascular tissue anyway? It is a special type of tissue, and I'll get to it when I get to the next group that does have vascular tissue, that forms a circulatory system within the plant. Now, this allows water to be transported from the roots up to the aerial parts of the plant. It also allows photosynthate to be transported long distances from the leaves or the areas of photosynthesis to the roots and so forth. And so the possession of vascular tissue has allowed these plants to become larger. All of the bryophytes are very, very small, okay? They don't attain a very large stature. They're usually mat-like, usually many individuals growing very close together, and none of them are very tall at all. So no vascular tissue, and that's the reason. So since they have no vascular tissue, they have parts that look like roots, parts that look like stems, and parts that look like leaves. And a lot of times these are actually called, just for simplicity, roots, stems, and leaves. However, since they don't have vascular tissue, they really are not true roots or true stems or true leaves. And so there's special words for these. The roots would be rhizoids, the stems are caulidium, and the leaves are phylidium, or phylidia for plural, phylidium singular. We'll keep the rhizoids, but I'm not going to worry about these, okay? I will never ask you about these terms. Just know that their stems and their leaves, due to the lack of vascular tissue, are not true stems or leaves, but we'll still retain the rhizoids. The rhizoids do not have any absorptive properties. They are usually just anchorage. All right? Waxy cuticle, that's an apomorphic for the plants, period. Okay? And but one of the things is that these, due to their reproductive structure, they require a film of water, or else they cannot reproduce. And that's why you usually find these groups in moist areas. Doesn't mean it's moist all year long, but it is an area that retains moisture for a good period of time for them to be able to reproduce. Type of gametangia, two different types. One is called antheridium, and that's a male structure. It produces sperm by mitosis. And archegonium, that is a female structure, analogous to ovaries, all right? So where mitosis, not meiosis, mitosis undergoes to produce eggs, all right, in their life cycle. In the bryophytes, in each of the three groups, the gametophyte generation is dominant. Now, what do we mean by dominance? We mean that that is the major photosynthetic form, okay, and that the sporophyte is dependent on the gametophyte for its nutrition, right? So the gametophyte is the main plant. The sporophyte is only used for further reproduction, right? So here are what they look like. This over here on the upper left is a hornwort. You don't come across those very often. Over here at the upper right, this is moss. In fact, this is Polysticum, the genus Polysticum. And this shows both sporophytes. Sporophytes is the green part down here. And the yellowish structure here is a sporophyte. And I'll come back to that. And this here is liverworts. Okay, and this is a close-up of a hornwort, which we're not concentrating on. But as far as the reproductive structures, usually this is what they look like in all of the groups. In one way or another, they come very closely resembling these structures here. So this here is an archegonium. And the archegonium consists of a venter, sometimes a little bit of a stalk, and then something that's called neck cells. And in this venter, okay, V-E-N-T-E-R, it'll produce one large egg. So this right here is the egg. And in it, you can clearly see the nucleus. It's the nucleus of the egg. And so in an archegonium, by mitosis, it'll produce just one large egg, okay? This stuff over here, this is sterile sheath cells called paraphyses. So in the lab manual or in the lab exercise, I have that word. In fact, it's going to be easier for me to write it. Where is it? Okay, so those sterile jacket cells, they're there for protection. They just protect the archegonium, and they're called paraphyses. They're not involved in reproduction, just protection. Okay, and so this is what, and from moss group, or from bryophyte group to bryophyte group, usually the archegonia sort of always have this sort of base-shaped structure, where there's a venter down at the base and neck cells up here. This over here on the right is an archegonium, okay? Or not antheridium, sorry about that. Antheridium. So these are the sterile cells up here that you see. And these are sperm cells. And so it produces by mitosis multiple number of sperm cells. These sperm cells have two flagella. We've seen that theme all the way through the eukaryotes, two flagella. All right, so this is what the structures happen to look like up close. I went over the life cycle of the moss yesterday, but I'm going to go over it again, okay? Because I really want you to understand these life cycles. And so the gametophyte is green, photosynthetic, okay? And this is one individual. Here's another individual. A lot of times, some individuals are joined at the rhizoid area. The rhizoids are down in the dirt, or in the substrate, whatever it happens to be growing on. And so this is the gametophyte. Has these leaf-like structures. That's where photosynthesis takes place. At the very top, in fact, if we go back, I don't know if I have a good picture. Do I have a? No, you really can't see it there. I may have a picture later on that shows it. You have at the very top, this is where the reproductive organs happen to be, the gametangia. And so it's at the top of the gametophyte. And if it's a male, so in moss plants, they're separate males and separate females. And so at the top of a male plant, it terminates in what's called an antheridial head, which contains numerous antheridia. And so all these little brown structures that we have right here, that's one antheridium, another antheridium, and so forth. And so this top here is not the antheridium. It is the antheridial head, and it consists of many hundreds of antheridia, or at least tens of antheridia. Paraphyses are these green things that you see right here in between the antheridium. Okay, they're there for protection. So it's in the antheridium that mitosis will produce haploid sperm cells. In the archegonial head over here, it's in the female. And once again, it consists of several archegonia, not just one, all right? So this whole structure here, even though it's not labeled, this whole structure here is the archegonium. And the archegonium consists of swelling part down at the very base called the venter, V-E-N-T-E-R. Within which it'll produce by mitosis a single egg. The egg is relatively large, and then the next cells appear. And then, of course, these green structures right here, these are the paraphyses, okay, which we've already gone over before. All right, so sperm is produced in the antheridia, and in the archegonia, they'll produce an egg. And in order for fertilization to take place, those sperm cells have to swim through a thin layer of water, moisture on that is uniting the gametophytes and the male and female gametophytes. This is why they grow together in real dense clusters, mat-like. If you really get down and look at moss, you'll see individual plants. And I'll try to dig some up, go find some because they're around, and I'll bring them into the lab on Monday so you can see for yourself what they look like. So we have these plants that are real close together, and at some time, usually in the spring or winter when rain has been around, there's a layer of water there. The sperm cell will swim by flagella. It has two flagella, has to swim to the female. And what it'll do is this sperm cells will enter the next cells, okay, down this tube where they meet the egg and fertilize it. We get fertilization, and we now have a diploid zygote, and it is retained in the archegonium. It stays there. It'll start dividing by mitosis, and now we have a embryo. The embryo is a sporophyte, and the sporophyte is still attached to the gametophyte. It'll keep growing by mitosis, and it has a completely different morphology. So this is attached to an archegonium. Even though there's multiple archegonia on the top of this archegonial head, usually, and many of them might be fertilized in the end due to competition of resources from the gametophyte. Only one sporophyte will arise. Okay, just one will win out. And so we have just one sporophyte, and it looks quite different, and it is not photosynthetic. Now here it has a stalk, and then a capsule. Up here is the capsule. So when you're doing your lab, and I'm asking you to label the gametophyte or the sporophyte, you would be wrong if you just only label the capsule. The capsule is only part of the sporophyte. It also includes the stalk. All right. So this is sporophyte, and up here we have diploid inside of the capsule. This capsule is the sporangium. This is the sporangium. Okay. And it's in here that diploid germ cells will then undergo meiosis to produce spores. As it dries out, there's usually with the moss, there are particular structure down here that will break open and that will release the spores that can be windblown. Okay, they can be windblown, and they'll end up anywhere. Most of the spores have a dead end because they end up on a rock, they end up on dry soil, but some of them will end up on moist soil where they will germinate by mitosis and form a protonema. The protonema looks very algae-like, like a filamentous algae. Then those will eventually become the rhizoids and sends up a bud, which will grow up into either a male or the female gametophyte. And that completes the life cycle. All right. Any questions on this life cycle?
You were saying that the capsule itself is the sporangium, and that's what can you repeat that a little? The capsule is the sporangium?
Yes, the capsule is the sporangium.
But the two terms can't be used for the same thing, right?
In this class, we can, because there's more to the capsule than just sporangium. Actually, more technically, the sporangium is a chamber within the capsule, but it takes up most of the capsule area. And so, in this class, I have no problem with using those interchangeably.
Thank you.
Someone else was asking something. Who else? Thought I heard another voice other than.
Oh yeah. Sorry, I put it in the chat. But you said that when the sporophyte it dries up, it releases the spores.
That's right.
Okay, thank you.
Yeah, because at the very end here in the capsule, that's more details than what we want to go into. And in fact, let me add something else. A lot of time, this figure here is showing a later capsule, but the very beginning sporophyte will have this sort of hat-like thing on here called a calyptra. I want to put this calyptra in here because that's something I do want you to know. Say calyptra, okay? Sort of looks like a hood, and this is in the early part of its development. Now this calyptra is actually haploid because it is, although it's on the gametophyte, it originated from the neck of the archegonium. So as the sporophyte starts growing, this neck piece tears off of the archegonium, and it closes and grows a little bit and becomes a little bit of a hat. And I think I have a figure here later on that'll show that. And so as it matures, first of all, this calyptra will be lost, and we have this state here. And inside just the opening of at the end, well, there's another cap-like thing, but as it dries out, because the water supply will eventually be cut off, and as it dries out, this cap pops open. And inside here, there's these teeth that go together. Okay, we call them teeth, but there's as they dry, they open up, and that leads to the release of these spores. Yeah, and so that has to dry out in order to release those spores. And the gametophyte will continue. Okay, the sporophyte dies, all right? And of course, the sporophyte, it is getting its nutrition, all of its nutrition from the gametophyte. It is completely dependent on the gametophyte. Now, there are a few cells here in the stalk, which is also called seta, S-E-T-A. It's a shame that one of the problems with botany is that there are so many words that have been invented for the same structure. And you're going to come across all of those. So stalks, seta, same thing. Some of those cells can be photosynthetic. However, they do not produce enough photosynthate to keep the sporophyte alive. And so the sporophyte is really dependent on the gametophyte. And this is why we say that in the moss or in the bryophytes, the gametophyte generation is dominant. Okay. All right. So looking at some pictures here. Yeah, this is real good here because this is a mat of gametophytes. All this here is antheridial head. So you can tell that is a male. That is a male. This here is an archegonial head. That is a female. And it's pretty typical that what you'd have is female, there's another female, that they're surrounded by male, okay? Because these sperm cells don't have to swim too far, okay? Because they're incapable of it. Under the microscope, okay, this is archegonial head. This is an archegonium. That's the egg. This bottom part down here, you don't see, it's not showing too much of the neck. You have part of the neck up here, but this down here is the venter. Okay, and if I go over here to the far, okay, let me get some things out of the way so I can see here. This here, you can see the sporophyte. And so when you trace the stalk down and see what plant the neophyte that the sporophyte is attached to, you know that is a female. Okay, you'll never find sporophytes attached to the males. Now this here, you can see this sort of pointed little hat piece up there, still yellow, right there. That is the calyptra. That is what comes off first as this thing starts to mature. Then over here, this is showing the end of the capsule of the sporophyte. The calyptra is gone. It's already been lost. And the cap has been sort of popped open, and you can see these interdigitating tissues that sort of look like teeth. They have been opening up, and this is where the spores will be released. This is a spore, and once that spore germinates, this is a protonema. It looks under the microscope very much like a branching green alga filament. Okay, so these are some images that make sense. Okay, and that's all we need to know about the bryophytes. Now, for the rest of the phylogeny of plants, let's just jump back here. I should have hyperlinked this, but from here on, this is a clade monophyletic that's called the Tracheophyta. And I don't have that word right here, but still the Tracheophyta is from here on up. It includes the Monolophyta, Gymnospermae, and Angiospermae. Okay, Tracheophyta. These are the vascular plants. They have vascular tissue, and that is the apomorphic, okay, synapomorphy that holds them together. So they have true roots. The roots are not like rhizoids. They are absorptive, okay? And they have true stems that have vascular tissue and leaves with vascular tissue. Now this vascular tissue it comes in two types. I don't know if I have it up here. Do I have it? No, I don't. But let's put it up there. I should have put it in here in the slides, but vascular tissue, two types, xylem and phloem. The xylem transports water and minerals, and usually the direction of transport in the xylem is from the roots to the leaves, okay? The phloem is another transport system, and it's usually right next to the xylem, and because the xylem and phloem are in what are called a vascular bundle, they're put together in what's called a vascular, and we'll examine this later on in the class when we get to plant physiology. So the xylem transports water and minerals from roots to leaf, and the phloem, it transports organic material along with water, of course, because it's in a watery medium. And that direction depends. It comes from source to sink. Okay, I'm getting a little bit ahead here. Okay, so the source is whatever is producing the photosynthate. Obviously the leaves, okay, because that's where most of the photosynthesis is taking place in the plants, is in the leaf. And so that's the source. The sink is whatever part of the plant happens to be requiring that photosynthate. And so it's growing tips, okay, where the plant is growing, actively adding tissue. Obviously that needs the energy and the material. And the roots are not photosynthetic, okay? They don't have any way to produce photosynthate, and therefore the roots are always a sink. And furthermore, so the flow is always from source to sink in the direction sometimes, like during the summer, a lot of times it's from the leaf to the root, or it's also the leaf from the, if it's a flowering plant, leaf to the flower, okay? But then during the winter or early part of spring, some of the flow can be from root to leaf because deciduous trees, for example, or deciduous plants, they lose their leaves during the winter and they have leaf buds behind, which is embryonic tissue. But then to grow out into a leaf and become photosynthetic, it needs material, it needs energy. So the roots store a lot of this photosynthate, okay, as starch. And then during the early spring, only in the early spring, you do get flow in the phloem from the roots up to the leaves until they become photosynthetic. Okay, nonetheless, it's a transport system, all right? And that's getting a little bit ahead. It's usually stuff that I reserve for later. So those two types of tissue, xylem and phloem. Now the xylem is also skeletal. And so one of the features here in the xylem is that they have special types of cells called tracheids, okay? All vascular plants have tracheids. As we get to higher vascular plants, the angiosperms, they have something in addition. Now what these tracheids are, they're long cells, okay? They're really long, all right? And they're dead at maturity. Okay, so as they grow, they lay down two thick cell walls, and then and those cell walls are pretty strong. Cell walls are pretty strong, so and they overlap. Okay, so this is showing one tracheid in the drawing, and then there's another tracheid that will interdigitate with it, and these will form long tubes. Where the two ends of the cells meet together, the cell walls are perforated. Perforated so that water can freely move from one cell to the other. Okay, now this water, and we're not going to worry about what is the force that drives this water. It's no pump. It's something else. It's evaporation at the top of the plant, and we'll get into that later. It's not important here. So these cells are dead at maturity. Okay, they constitute wood. Whenever you refer to wood on any plant, we're referring to the xylem. So it's both transport system and also skeletal system because of the thick cell walls. We'll talk about secondary cell walls a little bit later on, but they provide mechanical strength because if you're going to take a little plant that originally was a moss plant and get it bigger and bigger, it's pull of gravity bring it down. So they have to have some sort of skeletal structure that keeps it up in the air as well. And so these are for mechanical strength as well. Furthermore, in the sporophyte, the sporangia are branched. Okay, now as we saw in the moss, we had the sporophyte had just one branch, one capsule, one sporangium. But we'll find here the sporophytes will have branched sporangia. That is typical of all the tracheophytes. And in this case, the sporophyte generation is dominant. And this evolution going from predominantly gametophyte to predominantly sporophyte generation being dominant is an adaptation to the dry conditions on land. Okay, most of these plants, the bryophytes, they're due to their reproductive structures of the gametophyte requiring flagellated sperm to swim to a female gametophyte has limited them to very moist conditions. But here, using the sporophyte as the main reproductive generation, which produces spores that can be disseminated in drier conditions, this is more of an adaptation toward terrestrial conditions. And we'll examine that as we go on here. All right, now there's one group here that I'm not going to talk about, but it's mentioned in your textbook, the lycophytes. These were the first land plants, or at least resembled pretty much the first land plants, and they dominated about the mid part of the Paleozoic. We still have a few around today, sort of a relict group, but I'm not going to talk about them. We'll go on here to the next group, which is the Monophyta, the ferns and horsetails, concentrating mostly on the ferns. I'm not going to give any anthropomorphies for this. This group was very hard to work out, because they've been around for a long time, since the mid Paleozoic, and used to be the horsetails and whisk ferns and bonafide ferns. Their relationships, they're put in totally different phyla. With the molecular techniques now, the molecular data indicates that this is a monophyletic group and it involves horsetails ferns and whisk ferns. All right, so I'm not going to give any apomorphies here for it because but the monophyly is supported by molecular data. That's all I would say. Okay, now their roots, and for this group, are usually adventitious roots. Now, when we're talking about root structure, roots can come in three basic categories. They can be tap roots. They can be, like here, advantageous. Advantageous, what is the word? Go back here. Adventitious. Adventitious, or they can spread. Right? It's not the technical word for it, but we'll get to it a little bit later on. So what a taproot is. Actually, I will go ahead and give the proper word here for it. Fibrous. Fibrous roots. Okay, so with a taproot, if you have stem of the plant with its branches and everything, and I'll put in the leaves as the ground, is that the root has one major axis that goes deep into the ground. Then you have other branches of the roots that are minor that come off of it. This is a taproot. Fibrous roots, stem, go ahead and let this be soil. The root system is there's no dominant major root. All of them are about the same thickness and they branch all over the place. So these are fibrous roots. This is sort of like what you'd see if you pull up grass. Okay, they have fibrous roots. If you pull up a dandelion, they would have tap roots. Now for adventitious roots are roots that are growing off instead of the bottom of a stem, but off the side of the stem. Because in this group, in the ferns, the stem is actually horizontal underneath the soil. So here's the soil. And so the roots are actually coming off like this. So the thick part I've shown here is the stem. And then the leaves, which are called fronds, they're of course coming from the stem, and they're aerial. Okay, and that's typical. Yes, Alcosim.
Is that like how they do grafting for trees? Like sometimes they cut a branch off and then they put it in the ground horizontally, or like even like a sugar cane, you could put it in the ground horizontally and it'll have stuff growing out of it.
It depends, because grafting it depends on the group of plants, because but grafting is very much like that. You have to, a lot of times with that, it's not necessarily a stem that's being grafted. It's part of the stem and at least part of the root. Okay, and then they can put that in the soil to get it going. And this is a little bit different. Okay, so if you're talking about gymnosperms or angiosperms, these plants have most plants have a lot of abilities to propagate vegetatively. Okay, so you can take off for example, a leaf and of a tree or even a tiny branch of it, take it home, put it in water so that the end of the stem will start growing roots, and then you can put that into the soil and it will grow and it'll develop normally. So that's not really grafting, it's transplantation. Grafting is frequently is used when you take one part of a plant from one species and then make it grow on the root system of another plant. They do this with grapes quite a bit up here in wine country, because there's certain parts of the plant, the upper aerial parts of the plant, No, it's actually the roots that get attacked by certain insects, the larvae of certain insects that happen to be in the soil. And so they've imported certain stocks, root stocks from France that are resistant to these insects. But we have the grapes that are produced. We want special kites for here in California. And so they've taken the top part of one variety of grape and fused it to the roots of the French variety and put them in the ground. Okay, and that's grafting. And plants can do that. You won't do that with an animal very easily. If you lost your arm, you can't take the arm of someone else who recently died and just tap it onto you and expect it to grow normally. Okay, does that clarify things?
Yes, thank you.
Okay, so back to the story. So these are adventitious roots that they have. And the leaves are called fronds, okay? And the leaves, of course, this is horsetail fern. And so here the fronds are a little different, all these green things coming off, but the typical types of leaves like of the ferns themselves, this is going into, okay, it's growing on wood, but there's enough soil down here that covers that wood. But in here would be a horizontal rhizoid. The rhizoid, or not rhizoid, it's called a rhizoid, but it's really stem. And what is growing out of it, this whole thing is the leaf. And the leaf consists of petiole and a blade. So the blade part, these are not leaves. They are pinnae or leaflets, but the whole blade from down here all the way up to here is the frond, the leaf, okay? And that's one leaf. This isn't a bunch of leaves. These are all parts of one single leaf, okay? Now the life cycle. The life cycle is still, we still find ferns in moist areas, and that's still because of the gametophyte, okay? But still the main plant you see growing there is the sporophyte. And this is where we say that the sporophyte generation is dominant because that's the long-lived plant. The gametophytes live for a very short period of time. And so we have a situation that's flipped over from that of the bryophytes in terms of alternating generations. However, we still see that for a brief period of time, sporophyte, when it initially starts, is still dependent on the gametophyte. Now, in the next group of plants, we'll see that the sporophyte is completely independent of the gametophyte. In fact, it's the other way around. The gametophyte is dependent on the sporophyte. And here, both the sporophytes and the gametophytes are photosynthetic. But the sporophytes, for a brief period of time, are dependent on the photosynthete of the gametophyte in their life cycles. So we'll start with the major fern that you usually see. That is a sporophyte. And on the underside of the leaves are structures, clusters of branched sporangia. Okay, remember that for Tracheophyta, one of the apomorphies is that the sporangia are branched. And these are clustered together in a structure that's called a sori, or sorus. Sorus, U-S, S-O-R-U-S, is singular. Sori is plural, okay? All right, and so this is what contains the sporangia. And so this is a sporophyte structure. Now, in many cases, when you look at these sporangia, or this sori underneath on the other side of a vegetative leaf, if you look at the fern and look at the various blades that happen to come up, some of them have sori, some of them don't. And so there are strictly some leaves that are just vegetative, meaning that they're engaged just in photosynthesis, whereas others are reproductive, and they are also photosynthetic, but they produce sori as well. So a lot of times this cluster of sporangia, they're protected by a sterile tissue that's called an indusium. Okay, so I've showed this before. This here is a sorus. And in this particular plant, they don't have an indusium. all those little tiny things here, if you really zoom in, all the little tiny dots in here are sporangia. Okay, and we'll get a close up here. So now this here is a totally different species. The whole thing here is a sorus. And right here, that vein, that is vascular tissue. So when you look at a leaf and you see these veins, those veins consist of xylem and phloem, okay? And sticking up. So this here is the sterol, right up here, all that on the inside. In here, that's the inducium. What you see sticking out the edges, this is a sporangium. So I'm going to take a closer look at the sporangium. This is a cross section, okay? And it's a drawing. I got it from a different book, so you can see it from side view. So this is the leaf up here, and this is the major stalk of the sorus, and we have branching off of this stalk are the sporangia. So that's sporangium. Here's another sporangium, and this right here is the sterile protective layer, the inducium. Going on, if we zoom in, now we're zooming in on one of the sporangia, and this is an early sporangium. Okay, here's the stalk down here, and the actual sporangium, what produces by mitosis, the spores are inside this multicellular structure. It's inside there. Going around like a mohawk haircut, as I said yesterday, is a layer of single cells, single row of cells that goes all the way around, called an annulus. These are thick cell or thick walled. This right here is a very thick wall. That's a thick wall. This is a thick wall. Whereas the outer wall is relatively thin. And in this annulus on one side, there are two very thin walled cells. They're called lip cells because they look like a pair of lips right in here. Lip cells. Now what happens as after meiosis takes place and spores are maturing, the water supply is cut off, and as water evaporates, this starts to dry out, is that these cells are losing water. And because of these thick cell walls that are going down here and a thin cell wall up here, okay, this is a single sporangium, yes. This is one single sporangium. As the water evaporates, this inner wall sort of collapses, folds inward, like that. And when that does so, it is sort of creating a force that's trying to straighten out the annulus. As these cells dry out, there is pressure now because of this cell wall on the outside bending inward like this to pull all of this annulus into a straight structure. Since the lip cells are very weak, that's the point of weakness, this is where it'll break open. Breaks open right at the lip cells. And this is a more mature, later stage. Notice how the cell wall up here is indented like that. Those thick walls still hold it together, but because of this loss of water, it's creating a force to sort of straighten out this annulus, and that's what splits open the lip cells and releases the spores. Okay? So just go through the whole life cycle. All right, let's start with the sporophyte. Okay, this is a sporophyte. Yes, Aquazin.
Are these spores still considered myospores or no?
There would be myospores. Yes. I haven't been using, I think I may have accidentally used that word the other day, because when we get into fungi, you're going to find that there's really two types of spores. Ones that are produced by mitosis and ones that are produced by meiosis. And so here in the plants, all of the spores are myospores. They are produced by meiosis. It's going to be more complicated in the fungi because in the fungi, they can produce two different types of spores, sexually, and therefore they'd be myospores. And many of the fungus groups, those myospores are given special names when we get to it. which we will after the plants here. But they also produce mitospores, okay? Spores that are produced by mitosis, and that's pretty normal with especially some of the molds and everything, okay? All right, but here in plants, all of the spores are myospores. They're produced by meiosis. Okay, so starting with the sporophyte. That's the main plant you see. That's the one that can be pretty big. And I'm rather fond of native ferns, and so I have a lot of them that I've planted around my house. But nonetheless, on the other side, sorus. Here's the sori. And within the sorus are the sporangia. It's in the sporangia that meiosis takes place to produce haploid spores. These are then released, and if they fall on a wet area, they germinate, okay? And they grow into, first of all, the protonemia. It looks like a filamentous green alga, but then it will eventually grow into this single cell, okay? So it's one cell layer thick, not single cell, one cell layer thick structure. It's heart-shaped, and this is the gametophyte. The mature gametophyte consists of about between, because it really depends on what group of ferns you're talking about, between 2,000 and 4,000 cells, okay? But it is one layer thick, and they're always sort of heart-shaped. Now, these are bisexual, okay? They produce both antheridia. Usually the antheridia are here near the tip, the apex of the heart-shaped thing, or off on the sides, can be up here too. But near the cleft of the heart up here, this is where the archegonia are produced. Now, it really depends on the species. A lot of times you would have a bisexual mature gametophyte that will produce both archegonia and antheridia. But in other species, it's not that way, even though they're bisexual and they have the potential to produce both archegonia and antheridia. What you frequently have is that, let's go here, if I can, yeah, let's go down here. A lot of times what you would have in many species is whichever one of these matures first, you'd have a cluster of gametophytes on close proximity. And whichever one matures first will produce archegonia. And those archegonia will produce a chemical that diffuses out and will suppress archegonial production in the other surrounding individuals, which will produce then antheridia. Okay, this is very common too. So it's very hard sometimes, depending on the species of a fern, to find a gametophyte that has both archegonia and antheridia on it. It really depends on the species, but this is another pattern that happens quite a bit too. They still all of them still have the potential to become either male or female structures. But once the first one matures, it produces a chemical that suppresses archegonial production in the surrounding ones. They still have to be close together, still have to be a layer of water. The antheridia will produce by mitosis flagellated sperm, just like in the bryophytes, that has to swim to the archegonia, enter the archegonia, leading to fertilization. Okay? And so that's what happens here. Now the archegonia, now this is showing that all these are produced on the underside, okay, the ventral surface. This is turned over so you can see it in this drawing, but it's really on the underside. And it still has, the gametophyte has rhizoids, which are for anchorage, just like in the bryophytes, but not for absorption. This is photosynthetic, okay? It has photosystems and it can produce by photosynthesis, produce sugars and everything else it needs. But this here is showing the antheridium with sperm biflagellate. The archegonia is still sort of base-shaped, but it's still smaller than what you have in the bryophytes. You still have a venter, a neck, that's still there, but the sperm has to swim, digest its way through the neck, and it'll come in, fertilize, And with fertilization, we now have a diploid zygote, which grows into an embryo that is attached to the archegonium and getting all of its nutrition. Now, this is a sporophyte, and it's getting all of its nutrition from the gametophyte. And then it continues to grow, still attached to the gametophyte, sending up a frond that will start to become photosynthetic, and a rhizoid or root system that down into the soil. The gametophyte is still attached to it, okay, and is still providing photosynthate until this sporophyte starts to be photosynthetic itself, and the gametophyte will die off. So we find the sporophytes, although they have vascular tissue, they could live in drier areas than what the gametophyte would be at. They are still limited because of where they're limited to moist areas simply because they originate from the gametophyte. And the gametophyte has to be for reproduction, has to be in a moist area. And so this completes, these are all here, the sporophytes. Okay, and that's the life cycle. So this here is showing a germinating spore. This is a close-up of archegonia. You really can't really see the egg too much in these. It's hard to tell the early zygote, early embryo, and so forth. And these are immature sporophytes growing up, and then the mature sporophyte. Okay. Now, ferns, they're still fairly abundant. However, they're not as abundant as in the Carboniferous middle part of the Paleozoic. Now, prior to this, it was mostly lycophytes. And so lycophytes formed the forest. They weren't real big, but they're still big enough. Back in the Carboniferous period of the Paleozoic, though, we have trees. And we don't know that they're really, they didn't have, well, yeah, they did have vascular tissue because this is Tracheophyta, but there was a lot of fern trees. We still have fern trees, but not as abundant as what we have back in this area. And a lot of our coal deposits are formed from swamp-like areas that mostly had these fern forests. Ferns are not as abundant today as they used to be. Okay, now this is showing once again a cross-section of a sorus, but upside down. These are the leaf is actually down here. And these are the sporangia. You can see the annulus right there. Okay, so that's enough about the ferns. The rest of the tracheophytes belong to a clade called Spermatophyta. These are also collectively called the seed plants, okay? You notice here that in the reproductive cycle of the ferns, there are no seeds. And therefore, the ferns and the bryophytes, they're collectively called seedless. And this is the type of thing I was talking about before with the Linnaean classification, often led to classification where we had a monophyletic group and a paraphyletic group put together. And so in older classifications, sometime back, I won't give the names of them, we had this division that seedless plants were put together as a group, and then all the seedless plants is a taxon as well. Well, obviously, the seed is an innovation, and indeed, it is an anthropomorphy for the Spermatophyta, but that leaves the other group, the seedless plants, as paraphyletic. Okay, but here in the classification we're using here, we're not using seedless as a taxon. It's just that's what they are. They don't have seeds, but it's a plesiomorphic condition. All right, so, and there's two branches of Spermophyta, and these are the Gymnospermae, which contain three different clades that are in a Linnaean classification phyla. So Gymnospermae is not a Linnaean phylum. This contains Gymnosperms, Cycadophyta, Ginkgophyta, and Coniferophyta. We're mostly familiar with conifers, and I'm going, although I'd like for you to know cycads, ginkgos, I'm mostly going to be concentrating on conifers here. But we have three groups that belong to Gymnospermae. The angiospermae are flowering plants. Today, they are the most dominant plant. Most species out here that you see happen to belong to the flowering plants. Okay, so these are more adapted to both of these groups. So I'm talking about both gymnosperms and angiosperms as spermatophytes. That they are really more adapted to terrestrial conditions in their reproductive cycles than even the ferns. Because here the gametophyte now has been reduced even smaller. And we have a reversal, a complete reversal, in that the gametophyte is retained inside the sporophyte and is completely dependent on the sporophyte. In the ferns, both were independents. Okay, gametophytes were photosynthetic, and the sporophyte depended on the gametophyte for a little bit until it became the dominant plant. Here, the gametophyte is totally dependent and reduced very small into the sporophyte. And that's an apomorphy. Okay, that's an apomorphy for spermatophyta. Furthermore, instead of having sperm that do produce sperm and eggs, we still have that in life cycle, but the sperm does not have flagella, nor does it have to swim through a layer of water. What has replaced that is something called pollination. The pollination contains the pollen. We'll get to this. I'm jumping ahead here, but the pollen is a reduced gametophyte, a male gametophyte. It will contain sperm, but it can be windblown or it can be transported by insects. And so really what's happening here in the reproductive cycle of both of these two groups is that the male gametophyte, which produces a sperm, is moved by a mechanism that doesn't require water. Okay, and that is really allows these two groups to spread into the terrestrial habitat. Okay, so let me go back here in the chat. Are ginkgophytes the plants which produce really stinky fruit? I don't know. They don't produce fruit. Okay, that's the wrong word. Okay, they don't produce fruit. So I'm not sure what stinky fruit you're referring to, but it can't be ginkgos. The ginkgos produce cones, and I've never really ran up to a ginkgo to sniff it, and so I don't know whether that cone stinks or not. But I doubt that this is the plant you're talking about. Okay, but if you know what it is, send me a picture. Sometime I will be able to figure it out. All right, so we have several apomorphics here. The reduction of the gametophyte, so it's totally contained within the sporophyte, pollination, reproducing swimming sperm, but also another entity called a seed. Seed is the embryo sporophyte. The embryo sporophyte packed in a structure that contains food reserves. Now we saw in the in the ferns, that for that sporophyte to get its start, it had to be attached to the gametophyte, and it was dependent on the gametophyte until it could put out leaves and be photosynthetic. Well, you see, we also still have that problem here with the seed plants too. That little embryo that is inside that seed still has to have some sort of reserves for producing its structure, and also for cellular respiration to provide the ATP for it to grow until it can become photosynthetic. And that, instead of coming directly from the gametophyte and being attached to the gametophyte, comes from food that is stored in the seed. This is one of the reasons that we eat seeds like peanuts and corn and so forth, is because those are packed with starch, proteins, and other fatty acids, other supplies that are necessary for the embryo to grow. The embryo there is very, very small inside of a peanut. Okay, and this is also has aided in the spread of seed plants because this is also a dispersal mechanism. Okay, which I'll come back to a little bit later. All right, so these are the major apomorphies. And if we go back to our, too bad I didn't hyperlink. I hate to run all the way back, but let's go back to the phylogeny. Okay, so we're talking about right here. So this is spermatophyta. We have seed, what it is, pollination, and extensive reduction of the gametophyte into the sporophyte.
Gonna be on Monday's quiz.
Why does today's lecture seem so long?
I know, right? He's going so fast.
Okay, all right. Now, before I go into the spermatophyta, I need to talk about the life cycles here, because with regard to the production of spores, there's two different strategies that are used in plants. These are homosperous versus heterosperous life cycles. In the spermatophita, they're strictly heterosperous life cycles. In some of the ferns and further back, we have both types. But here, what we have with the homosperous sporophyte, homosperous sporophyte will produce one single type of spore. All right, and so this is very much like this is what happens in ferns, right? One type of spore produces when it germinates, it produces a bisexual gametophyte that has both archegonia and antheridia, and therefore produces both sperm and eggs. Heterosperous sporophyte. This is like in the moss that we have discussed, and also strictly in the spermatophyta. This is heterosperous cycles. It produces two different types of spores. One that's called a microspore, one that's called a megaspore. Now, this microspore megaspore distinction in moss is not usually pointed out. Sexually, there are two different types of spores. One that when it germinates will produce a male gametophyte, one that will produce a female gametophyte, but the two types of spores are not morphologically distinguishable. But here in the spermatophita, that they are, okay, they look different. So with the microspore, when it germinates, it produces a male gametophyte, which is also called a microgametophyte. In plants, we have duplication of terms. So a male gametophyte, microgametophyte, same thing. I prefer to say male gametophyte, all right? So we can keep the sexes straight here. The megaspore will germinate into the female gametophyte. Now the male gametophyte, therefore, will produce sperm, whereas the female gametophyte will produce an egg, okay? So this is the heterosperous sporophyte. Two different types of spores, okay? All right, so I'm going to look at each of these two groups. First of all, gymnosperms. Okay, so I put up here abbreviated lineage. Our lineages are getting bigger, okay, as we're going on here. So this would be Eukarya, Archaeplastida, and what else would go in between here? I've left some Viridiplantae, and then Plantae, Tracheophyta, Spermophyta, Gymnospermae. Okay, now Gymnospermae, three different clades. We're mostly going to be concentrated on one group. So the reproductive, I'm putting it as an apomorphy. One of the reproductive structures here in the gymnosperms is something called as cones. And these cones, that is on the sporophyte. And we'll look at how those cones work here shortly. So that's a sporophyte structure. And of course, sporophytes, because these are tracheophytes, it's dominant generation. And this includes three different clades. Ginkgos, in which there's only one species alive today. Used to be several species in the past. Cycads, which is fairly numerous, and conifers. Conifers are the biggest group. It's the biggest group that we have today. And there's one other group that we don't need to worry about. They're sort of strange. Okay, so this is cycads. So the official name here is Cycadophyta. You can just refer to it as cycads. I don't care. You don't have to know the scientific name. So they initially sort of resemble superficially to a certain extent palm trees, which they're not palm trees. And this is a cone. All right, so that's the reproductive structure. And it's very similar to what happens in cycad cones are very similar to what happens in conifer cones. I'm not going to give any apomorphies for this group, but you should know that it's a clade within the gymnospermae. Then there's ginkgos. The ginkgos also produce a cone, although it doesn't look like a cone. Okay, this is a male cone, female cone. Okay, actually, those are seeds that will develop from the cone. Now, this is the leaf of modern ginkgo tree. Ginkgos were thought to be extinct. We had fossils of them. The leaves are quite distinct like this. And it was found, I don't remember when, sometime in the 1800s, I believe, that ginkgo trees were found in a monastery in China. And people were very surprised, excited, because ginkgos were not extinct after all. So we only have one species that is alive, and that species now has been, planted in other places. Yeah. So is the same with Ginkgo bilobata. Yeah, that's exactly the name. That's the genus and species name. Okay, Bianca, you have your hand up.
Oh yeah, this is what I was asking about because I think one of the ginkgo trees produced some kind of fruit, and then when they were crushed, they would produce like a really bad, like vomit-like smell. And I was wondering if this was related to.
I will look into it. Fruit is not the right word, though. Okay, say yeah, because as we get on a little bit later, when I get into the flowering plants, that's where you find structures that are called fruits. Now, colloquially, very often people will use non-scientific words for something that whatever they think it is. But this group does not produce fruit. But I don't know. I've never. I'll check into it.
Okay.
And this may be what you're talking about. I've never taken any of these cones or anything or the seeds and broke them open to see what they smell like. Even though there's a ginkgo tree not too far from my house. So when I walk the dogs, of course, it's not producing anything right now. Anyway, I'll check into it. This may be, but Ginkgo biloba, that is the name of the species, genus and species of this one survivor. And I have a couple of ginkgo trees growing on somewhere down the street in my neighborhood, and on the campus of the UC Berkeley, they have some ginkgo trees up there, but they're not native to here. Okay, these came from a Chinese monastery, so we now have this group around. Now, the major group of angiosperms are the coniferophyta, okay, conifers. And this is typical needle leaf trees that we see around: pine, spruce, redwood, larch, fir, junipers, cedar. These all belong to this group. Most of them are evergreen. When you say evergreen, they lose their leaves all year long and grow new ones. Okay, but they don't typically lose them in winter time in temperate regions. This group was really hard to come up with the morphological character that typifies it as an apomorphy. Genetic information has resoundingly put all the conifers together as a monophyletic group, and also molecular data has put together the gymnosperms as a monophyletic group. But I'm going to put here that they have needle-shaped leaves. Okay, they don't have the broad, flat leaves. Now, it is indeed that junipers and cedars, their leaves are somewhat flattened, but they're still not broad, flat tissues like we find in angiosperms. And so we'll go ahead and just give this as an apomorphy so that we have something to grab onto. Yeah, Aquasim.
Aren't redwoods like a type of fir? Isn't it called like a redwood fir or something?
A redwood fir? Or no, well, there's red fir. I've never heard of a redwood fir. But yeah, there's different types of fir trees. There's white fir, red fir, and there's a bunch of other types of firs. Is that what you're referring to?
Yeah, I might have got them confused.
It's not called a redwood fir. It's called it's called red fir. Okay, and Marielle.
Can you explain evergreen again?
Okay, most of our oak trees, and in fact, around here we have several species of oak trees and other types of trees that in temperate regions, when conditions for growth in winter become bad, is that they lose all their leaves. Okay. But we have conifers, they retain their leaves. Even in winter, they still have the needles. Okay, they don't drop them. You don't go out there and find just, with our redwood trees, you don't find during the winter just bare limbs. All right, that is evergreen. That means they have leaves. They keep leaves all throughout the year. Okay, now this being evergreen is not restricted to just these conifers, because as I said, we have some oak trees around here that retain that coastal oak tree. It's called coastal live oak. We also have canyon live oak, and they keep their leaves during the summer or during the winter. You can see them right now. You can just, if you're on campus and look up at the hill, you still see trees there that have, other than those eucalyptus, if you look further up the hill, that there's those are coastal live oak. But you look around too, you can find some oak trees here that they're barren at this time of year. They've dropped their leaves. That they don't have any leaves. So valley oak is like that, blue oak, and what other oak do we have here? Those are the main ones. Oh yeah, black oak, we have that too, that species, and they drop their leaves during the winter. You just go out there and you just see bare limbs. Does that clear it up?
Oh yeah, thank you.
Yeah. Okay. And so these needle-shaped leaves are sort of adapted to dry, and of course vascular tissue. Obviously, the tracheophytes. So there's tracheids. They have those cells, and those are in the xylem. Once again, I mentioned water transport, mechanical strength. If you know the phylogeny, you don't have to really say that because it occurred further down the tree. And so those aren't apomorphs. Some of these are very long-lived. Okay, so there's a bristlecone pine that's growing in the mountains between California and Nevada, and its location is kept secret. It's the oldest living tree we know of, and it has been there. We know because you can drill into these trees, because every year they lay down a new layer of xylem, and xylem is wood, the wood part, and they're in rings, and so growth rings, we can count them. So this critter is 4,600 years old. It's been around for quite a long time. There was one tree that was earlier in the 1900s, there was another tree here that was older than this in the same area. And a graduate student from UC Berkeley back then was wanting to study it. He went out there and he chopped down in order to count the rings. And so to protect this tree, the Forest Service protects its location so no one can harm it. But anyhow, that's oldest tree. Oftentimes, these conifers are growing in dense stands of the same species. And so this is Douglas fir. This is more up in Canada, or could be up in Washington, because they have usually big stands of fir trees. And there's a reason for that too. And it's sort of analogous to the close growing conditions of bryophytes, but for a different reason. And I'll get to that a little bit later. Redwoods, okay? We have two species of redwoods, and actually they're in two different genera, but they're still in the same plant family. One is the coastal redwood, which we have growing even here in Fremont, Danville, and other places as an ornamental tree, but it's not native right now. Usually it's natively grows closer to the coast, over in the Santa Cruz Mountains and on up. But then we also have in the Sierra Nevadas, another, Sierra, it's a different species of, as I said, even a different genus. I think it's called Dendrosequoia. This is called sequoia. The one in the Sierra Mountains is Dendrosequoia, and they're both belong to this group. Okay, all right. So Aquazim, your hand is up. Is that new or is that old?
Yeah. I just looked up the bristlecone pine tree because I was interested. Is it not evergreen? Because it looks like it doesn't have any leaves on it.
No, they still retain leaves. Even during the growing season, they don't have many leaves.
Okay.
pine I've ever seen. They have very few leaves on it. And a lot of times the tree is not that big, even though it's thousands of years old. It's more like a bush. But I'll look that up too, because I'm not sure. I don't think that they lose their leaves, but I'll check it out.
Yeah,
Okay, now,
it looks like it's just like one like just the it has like I don't know how to explain it. It's just like no leaves at all on it. At least on the picture of what they are saying is the oldest tree, which is the Bristol cone pine.
well, the one oldest tree is a bristle cone pine, but there's a bunch of those. Okay, so that's a species, and so you would have population. Okay, but I'll check it out, because I don't think that they lose their leaves, but I may be wrong.
Okay, thank you.
So, as far as life cycles here, I'm only going to concentrate just on pine, okay? But it's still typical of a conifer life cycle. I'm not going to go over life cycles of cycads or others, but let's go over this. Okay, so, and this is going to be important for Monday's lab, all right? So heterosporous cycles. So we have two different, now cones, that is the reproductive structure on the sporophyll, or on the sporophyte. And we have two different types of cones. Male cones, which are called staminate cones. In these cones, they produce the microspore, okay? And they have, oh, do I have it here? I'll stick with it, finish up the staminate cone. Staminate cones are male cones. Only sperm or spores, microspores will be produced there. Okay, so these cones, what they are, is they're a cluster of modified leaves. Modified leaves that are put together in a cluster. And so these leaf-like scales that make up this cone are called microsporophylls, right? When you see this phyll, P-H-Y-L-L, that is a Greek word that usually refers to leaf. And so these are microsporophylls. This is where we will have a microsporangium that will undergo meiosis to produce microspores that will then be released. Okay, so it looks like I have a hyperlink here. Let me go to it. It's actually, yeah, okay. So this is not a male cone. This is a cluster of male cones. And so this right here is one cone, a micro cone, a male cone, okay, staminate cone. And you have all these scales on it, and each of the scales is a modified leaf. Now, if we take a cross section, actually a longitudinal section, we're going down through it, and this is what we'd see. Here's the middle stalk of the scale. This is a microsporophyll, a little bit of a leaf-like structure. That's the microsporophyll. And inside what constitutes most of the interior here, let me put it right, almost all of this inside is the microsporangium. Microsporangium. So the whole scale is a microsporophyll, and it's also called a pollen sac. This microsporangium is the interior of that microsporophyll. And this is where meiosis will take place to form microspores. Okay, so Aquazim, is it new hand or old hand?
Old hand.
You sound like you're in an echo chamber. Anyhow, that's all right. Okay, so that's the cone. Part of that microsporophyll. So microsporophyll equals the scale, and that one of those scales that form part of that male cone, staminate cone. Interior part called a pollen sac or a microsporangium. And what we have inside early on. So this is a developmental series that takes place inside of that sporangium. First of all, it is filled with microsporocytes. Okay, CYT cell. That's a suffix that refers to cells. These are diploid. Okay, they will undergo meiosis to form 4 microspores. Okay, 4 microspores. Okay, so this is a close-up of a microsporangium, real close. And we're going to have microscope slides very much like this when you come in on Monday. And so inside, so this whole thing here is the microsporangium. So that includes sterile tissue, okay, called an integument. The integument is all this stuff around, and then on the inside is the microsporangium. Okay, all of this, the interior part. And initially, this is filled with microsporal sites. They will undergo meiosis to produce haploid microspores. Okay, and those microspores, okay, it's a microspore, it will still germinate. It's not released. It germinates still in the cone. And it doesn't go through too many mitotic cell divisions. Now, once that spore has become 2 cells, it is an immature male gametophyte. It is the gametophyte. And in most species of pine, it only contains 4 to 6 cells. That is the male sporophyte, what we call pollen. So what a pollen grain is, and this is going to be true not only in this group, but also in the gymnosperms, but also in the angiosperms, the pollen grain is a male gametophyte, really reduced in size. Here, it's only 4 cells, or 6, depends on species. And so this is also called a microgametophyte, all right? I prefer that you really get close in your mind, really bring this together, that the pollen grain is a male gametophyte. I don't care if we use this word microgametophyte. So here we have 3 different terms for the exact same thing. Pollen grain, male gametophyte, microgametophyte. But let's keep it at male gametophyte is a pollen grain. I really want you to make that association. It's real important for understanding this life cycle. This wasn't appreciated until way, way late, okay? So when it's mature, and it matures inside of the sporangium, right? It usually these have sort of wings on it. I'll show a picture here in a minute. And what the function of that is that all pollen, all male gametophytes are distributed to female cones by the wind. They're all wind pollinated, all right? Wind. And therefore, these wings aid its dispersal, okay? No water. So this is a pollen grain. This is a male gametophyte. And these are the wing-like structures so that it can stay aloft in the wind because wind pollination is how it all goes. All right. So just before, I have just 2 minutes, staminate cone. Let's run through what happens. In this mature cone, pollen, okay, 6 cells or 4. There's 2 real important cells there when the pollen is released. Something called a tube cell and a generative cell. These are there in the pollen grain when it's released from the microsporangium. As this happens to dry out, the cone the microsporangium splits open and the pollen is released. Once again, the pollen is a male gametophyte. What's important about this generative cell is that when it divides, it creates sperm. And so the male gametophyte produces sperm, and that it needs to be carried to an egg that will be produced in a female cone. Now, I don't have time to go over the female cones now. I was hoping to get to that before we get into Monday. But as briefly at the beginning of Monday, I will complete this. Now, problem is that since this is wind blown, it's fully up to chance as to whether that pollen will fall on a female cone or not. The vast majority of pollen just falls on the ground or somewhere else. And when the pollen is being released by these trees, we're talking about millions and millions and millions. Although the pollen is very, very small, the energy expenditure on the part of the tree is tremendous. Really, really tremendous. Because most of this will go nowhere. Okay? And this is why they produce large amounts of pollen. Also, why conifers usually grow together in dense stands of the same species is because windblown pollen doesn't go very far. Okay, and therefore, the closer they are together, the higher probability of the pollen reaching a female cone where the female gametophyte with an egg will be located. And I'll go into how that works later. These are called, female cones are called ovulate cones, but this is where I'll stop. Okay, any questions?
Sorry, one last question. Is the microsporophyll the same thing as the staminate cone?
No, the staminate cone, let's go back here. You can see it. In fact, when you're walking out, looking around at conifer trees right now, it's on campus, look near the tips of the branches. You'll find a bunch of little tiny clusters of stuff like this. Okay, this is a cluster of staminate cones. Staminate cones are real small. Now, when you're talking about pine cones that you usually pick up and off the floor and everything like that, those big cones, those are ovulate cones. Those are female cones. Most people don't pay attention to these male cones. The male cones are very small. You always find them at the tip of the branches, not near the middle. The ovulate cones, the female cones, you usually find toward the middle of the branches, not out at the tips. But this whole thing here is a staminate cone, and it consists of several hundreds of leaf-like modified leaves. That is called the microsporophyll. Inside is a sporangium. And inside of that, early on, we will have microsporal sites, a cell, single cell, diploid cell. Diploid cell, microsporal site, that it will undergo meiosis. And of course, meiosis is 2 divisions that will produce 4 microspores. This is the microspore. And so it's been produced inside of a microsporangium that is inside of the microsporophyll, which is inside of or on the surface of a staminate cone. All right. And then these will germinate still in, but there'll be several cells that make it up, the male gametophyte, which is the pollen grain with its ears. Okay, does that clarify it?
Yeah, so there's no term for the whole bundle of those cones right there?
This right over there? No, it's, you just call it, there's no special term for it. It's just a cluster of male cones, cluster of staminate cones. They usually occur together in clusters like this, not individually. That's a little bit different with the female cones. The female cones are usually isolated, individual from each other, but they're not in clusters like this. But having the staminate cones clustered together at the tip of the branches, that's pretty common. Okay. All right. Any other questions?
Oh, just to clarify something.
Mm-hmm.
So after meiosis, it's still called microspores, right? The 4 haploid cells.
Okay. First of all, we have a diploid cell, and that is the microsporal site. When it undergoes meiosis, once we have meiosis, okay, meiosis, reduction division, then those are microspores. So the microsporal site after meiosis will form microspores. And then when the microspores divide by mitosis, they become at least 2 or more cells, then that is the microgametophyte. And the microgametophyte, I'm not emphasizing that particular word, it is the male gametophyte, which is also called pollen. Okay, does that get it?
Yes.
Okay, yeah, some of the stuff, probably make a flow chart. This is the best way to keep track of it and to learn it, and superimpose on this that generalized life cycle of alternating generations of plants, because it's still here. It's just the details are getting more intricate. but we still have alteration of generation with these sporophytes and gametophytes. Okay. Okay. Any other questions? Okay, then
Oh wait, sorry, I have another question.
sure, go
I'm
ahead.
sorry. For slide 37, I'm still a little bit confused how to differentiate like different species between like the homosporous and heterosporous life cycles.
Okay, let me go back to that. Let me end this. Makes it easier for me to get back there.
Like, is it homosporophyte like
Okay.
the fern?
Which slide was this?
37.
30.
7.
Yeah, there. Okay. That was cool. Okay, so there's just two different in plants in general, okay, because it happens in various groups here. What happens with the homosporous life cycle, it's referring to what type of spores are produced. Are these spores a single type of spore that produces both types of the gametophytes and produce sperm and eggs, or do they have two different types of spores? So homosporous, homo meaning the same, okay? Spores referring to spore. What we saw in the fern life cycle is they're homosporous, okay? So the sporophyte produces one type of spore that when it germinates, we have a bisexual gametophyte that can produce archegonia or antheridia and produce sperm and eggs, okay? So that's bisexual, even though, as I pointed out, some species of ferns, when we have a gametophyte surrounded by other gametophytes and it matures first, it only produce archegonia. It still had the potential to produce antheridia. They're still bisexual. They still have two different sex determinants genetically. That can, okay. With the heterosporous cycle or type, we saw that in moss. Okay, Polysticum, which was the main type of moss that I was using here as an example, they produce two different types of spores. However, morphologically, they look the same. But one of them, when the spore germinates, it grows into the male gametophyte, which only produces sperm, only has antheridia. It's not really in the, well, we'll go ahead and keep that term, megaspore. When it germinates, it becomes a female gametophyte with archegonia and therefore produces eggs. So the whole point, we have both of these, and it's not a phylogenetic thing. Some groups are totally homosporous. There's some groups that I haven't talked about that can be either one. But I'm introducing this, are examples that I covered in this class. The ferns, species of fern that I used here, is homosporous, whereas the bryophyte, the moss, heterosporous. But when we get into spermaphita, that is in gymnosperms, pine, and the angiosperms, we're only going to find this, only the heterosporous type.
Okay, that makes more sense. Thank you.
Okay. All right. Okay. Anything else? Okay. So do we stop at slide 49 for Monday's quiz? Yes. The last slide I went to. Let's see, where am I? I'm going the wrong way. Yep, this is where we stop. Okay, now where did we begin? What did I start on Tuesday? Let's see, I usually write it down because from year to year it's always something different. So I started on Monday or Tuesday. I started with with the super groups, so Apicomplexa. So we'll still start from that slide, Apicomplexa, up to 49 here, and that's the stopping point for the material on the quiz. Okay? All right. Anything else? Okay, then everyone have a good weekend, and I'll see everyone on Monday on campus. Right, we're going back to campus.